Welcome back to the biology series. So today we're going to continue with the CSEC biology and also the CSEC HSB syllabus. So basically we did the nervous system before, we did cell division, mitosis and meiosis, we did cells, animal versus plant cells, we did which one? What's it called? Cell structure and function. So we did, what is that, four? So we did those four already. So we're going to continue the biology series talking about diffusion, facilitated diffusion, osmosis, and also active transport. So that's the plan. Let us continue. Alright, so we're going to start with diffusion. So now we can either just call it diffusion or we can call it simple diffusion, and basically all of these are methods of transport within the cells. All of these help to get stuff across the membrane or cell membrane within the cell, basically, right? So diffusion, facilitated diffusion, osmosis, active transport, all of them function to get stuff across our cell membrane, right? So we can either call it diffusion or simple diffusion. It's the same thing, basically. And what is diffusion? So basically, diffusion is the movement of particles from an area of high concentration to an area of a low concentration until it's evenly distributed, right? Because that's the goal of um, diffusion, basically. The goal is always to get the stuff evenly distributed. And the example I learned when I was doing, I think, maybe integrated science back in probably grade 8 or grade 7, my science teacher at the time, he would say, for example, if somebody opened a box of KFC in the classroom, right? So the person that way down at the back of the classroom and them have some KFC, them eat for lunch. And when them open that KFC, you best believe, say, if you stand up at the door of the classroom and them at the back of the classroom, that smell, the KFC smell, is going to move from that area of high concentration at the back of the classroom to you that is right at the door, because that's how diffusion works. It's always to get the particles evenly distributed. So it's going to always move from an area of high concentration to an area of low concentration so that it can be evenly distributed, because that's always the goal of diffusion. Right, so that's basically our definition for diffusion slash simple diffusion. They're about the same thing, but just remember, high concentration to an area of low concentration, and the goal is always for it to be evenly distributed. It doesn't want, like, just everything bunged up in a one corner, right? So for example, you see these balls, these could be molecules for something, and they're literally concentrated in one area, right? What diffusion is going to do, diffusion is going to move it from this area of high concentration, and look at the entire, like, look at this container. Literally, when you look around it, it's all of it is empty. So it's going to move from an area of high concentration to an area of low concentration to get it evenly distributed, because that's always the goal of diffusion. Alright, so examples of substances that diffuse. So substances that use diffusion include water, oxygen, carbon dioxide, ethanol, and also urea. So these molecules can pass through cell membranes without energy through simple diffusion, right? And then another example would be the gases in our lungs, right? The gas exchange between oxygen and carbon dioxide. So during respiration, oxygen and carbon dioxide are exchanged in and out of the lungs through simple diffusion, right? So these are just giving you examples of substances that use diffusion, right? And then you have glucose. So glucose moves into cells through diffusion for respiration. And then ions also pass through the membrane, right? But they don't just go through the membrane just like that. They need help. So that's why another type of diffusion is what they use to get across the membrane. So they use facilitated diffusion, in which I'm going to get into more details about facilitated diffusion very, very soon. And then in the case of this glucose, what gets glucose across the membrane is, it's not necessarily just diffusion, right? Because glucose is a very big molecule, right? So for something like diffusion, another type of transport would actually get glucose across the membrane. So active transport, or even, yeah, I think active transport would be able to get glucose across the membrane. So when diffusion can be a very broad term, it depends on how you use it, but transport is more than just diffusion. It breaks down a lot more. So we're going to get into more details and explain all of this next. Right, so let's talk about the rate of diffusion. So the rate of diffusion is affected by several factors, including temperature. So temperature, concentration gradient, or surface area, like for example in our cells, or surface area to volume ratio. Also the diffusion distance, density. So the higher the temperature, so higher temperatures increase the kinetic energy of particles, causing them to move and mix faster. That means that the higher the temperature, the faster the particles are going to move across the membrane. The faster it's going to happen. And as it relates to concentration gradient, so the greater the difference in concentration, the faster the rate of diffusion. For example, you have like a lot of particles. So you have maybe we have 10, so we have 10 molecules outside of the membrane, right? That needs to get inside, right? Or something like that. It's moving from an area of high concentration, right, to an area of low concentration. You get what I mean, right? So when it says the greater the difference in concentration, imagine if, for example, you throw, like, you probably pour some water, right? You pour the water. When you have a lot of water in the bottle, for example, and you throw the water in one cup, it will move really, really fast. When there's a small, tiny bit of water left, because the concentration get lower now, it will take a longer while. So we don't know if that is necessarily a good example or that I explain what it should, but the greater the difference in concentration is the faster the rate of diffusion is going to be. As it relates to surface area, for example, our cells have a large surface area to volume ratio, so a larger surface area results in a faster rate of diffusion, right? So for example, our cells are so small, and because they're so small, they have that large surface area to volume ratio, and because they're so small, it means that they absorb things quickly, right? So if something needs to diffuse into our cells, it's gonna happen really, really quickly because they're so small, right? So the smaller it be, the faster diffusion is. And then another example is, for example, our root hair cells in plants. So they line, and also cells lining the helium in animals have increased surface areas to absorb nutrients, right? We understand why root hairs would need to, have a large surface area, because they need to absorb a lot of water. And also the ileum, instead of our, I think, small intestine, so the helium in our small intestine, it would also need to have an increased surface area in order to absorb nutrients, like literally. So it all makes sense. And then as we go on more to explain diffusion, right? Distance. So the shorter the distance molecules must travel, the faster the transport will occur. For example, blood capillaries and alveoli have walls that are only one cell thick to speed up diffusion, right? And the best way to explain this is, for example, if you're going somewhere, right? If you're going somewhere, let's just say if you're walking down the street, so you, or maybe you're going shop or something, but let's just say this: the shorter the distance, the faster you're going to get to where you need to go. The longer the distance, the longer it's going to take you to get where you need to go. So it's the same thing as it relates to diffusion and distance in our molecules. So the more dense a particular molecule or solution is, that means that diffusion is going to be decreased, and it's going to go a lot slower, because density would refer to like particles being packed extremely tight, right? they'll be very, very packed tightly together. So if they're packed so close together, it's going to take them a while in order for them to separate, and that means that diffusion is going to be hindered. So diffusion is going to take a longer while if, for example, the solution is very dense. Alright, so the next one is facilitated diffusion. So basically, both simple diffusion and facilitated diffusion, both of them are methods of passive transport. So both of them, so simple diffusion, facilitated diffusion, osmosis, active transport, all of them move stuff across the membrane, right? And then simple diffusion, facilitated diffusion, and also osmosis. Those three are methods of passive transport. The reason why they are methods of passive transport is because they do not require the input of ATP, they do not require any energy whatsoever. So if we're at facilitated diffusion now, and passive transport process that moves molecules or ions across the cell membrane from an area of high concentration to an area of low concentration. And so the biggest difference between facilitated diffusion and simple diffusion is that facilitated diffusion, the molecules need some help to get across the membrane. So in our simple diffusion, the molecules are able to just simply diffuse directly through the membrane because they could pass through the hydrophobic tails of the phospholipid bilayer. They could just pass through. In facilitated diffusion, these molecules are actually very they are hydrophilic. So if the molecules are hydrophilic, if they're polar and they're charged, they cannot pass through the membrane. So they need some help. So what's going to happen is that we're going to have specialized proteins in our membrane, meaning our protein channels and also our carrier proteins. So they're going to be embedded within the membrane, and they're what helps larger molecules, polar, charged molecules, and also molecules that actually cannot pass through the hydrophobic tails. Because at the top of the membrane, the heads are very much hydrophilic, but in order to pass through the membrane, they have to pass this area, which basically has the hydrophobic tails, meaning that this area cannot handle water, right? They hate water, they hate any kind of liquid, right? So they cannot pass through there. And so what these channels, so this is the protein channel and this is the carrier protein. So you can see the difference in the size. For the channel protein, it's basically like a hot dog. It's folded, it's shaped like a hot dog, and it transports the molecules directly across the membrane. In which now, on the other hand, the carrier protein looks a bit like a pump. It kind of resembles active transport somewhat. Right, so both of them are mechanisms that are both used to move stuff across the membrane. Right, so one is a bit more, round or, enclosed, kind of like a pump. And so basically it's like one side closes, one side open, taking what it needs, then it closes that side and then it opens here and then it lets out the molecules here, versus our protein channel where the stuff just passed through. Right, so that's the biggest difference between simple diffusion and facilitated diffusion. One requires some help, and these are our channel proteins and our carrier proteins. They're what helps to get these molecules across the membrane, because it's literally the same thing. In simple diffusion, they left from an area of high concentration to an area of low concentration. In facilitated diffusion, it's the same thing. If the molecules are moving from an area of high concentration to an area of low concentration, but this time they're going to use specialized proteins to help them get through the membrane. And examples of that would be urea and amino acids. Now back here we say that glucose moves into cells through diffusion for respiration. So glucose is a molecule that can either use facilitated diffusion, or it can also go through active transport to get those stuff across the membrane. So now this is a much more larger diagram, right? So basically this is the phospholipid bilayer. So this is the fluid mosaic phospholipid bilayer. This is our channel protein. This is our carrier protein that resembles a pump somewhat, and facilitated diffusion does not use ATP, right? So we have the hydrophilic head and we have the hydrophobic tails, and this part of the membrane cannot be, it's, what should I say, it's lipophilic, right? So it's lipophilic, meaning that it's fat, it's lipid loving, right? And if it's lipophilic, that means that it's hydrophobic. So certain molecules that are really charged, really polar, cannot pass through this area of the membrane. So they can come up here, but they cannot pass through in order to get inside. So that's the biggest difference. Okay, so specialized proteins that help move these molecules, so that's the difference. In our facilitated diffusion, we use carrier proteins and we use channel proteins. So carrier proteins, they bind to the substance being transported, right? And then channel proteins allow the substance to flow freely. So as you can see, it literally binds. Look at that, specifically shaped out for the molecule in order to get it across the membrane, while the channel protein just like lets them go through. Right, so facilitated diffusion does not require energy from the cell. It's used to absorb nutrients like amino acids and glucose across the gastrointestinal tracts, and also, the epithelial membrane. So now factors that affect facilitated diffusion. So factors that affect facilitated diffusion. So Brownian motion is the force behind the diffusion of lipids. So the main factors affecting the process of facilitated diffusion are temperature, concentration, diffusion distance, and also the size of the molecules. So these are basically almost like the same thing as the regular simple diffusion. So for simple diffusion, we said, okay, temperature increase, the movement of the particles also increase, so they will move faster. Concentration, the higher the concentration, that means that the molecules are going to move faster to get to the area of low concentration. Diffusion distance, the shorter the distance, the faster they will get where they need to go, essentially. And also, when we talked about the density, meaning that earlier, but in this case, we're talking about the size of the molecules. So the smaller and lighter they are and uncharged, they will move across the membrane a lot faster. So now we're moving on to osmosis. So this is the process where water molecules move from an area of high concentration to an area of low concentration across a semi-permeable membrane until they're evenly distributed. So osmosis is a form of passive transport, just like simple diffusion and facilitated diffusion, but the difference with osmosis is that it takes place specifically within water and it passes through a semi-permeable membrane, right? Semi-permeable means that some stuff get across, right? But yeah, so it's the same concept, high concentration to an area of low concentration, because the goal is always to have the molecules evenly distributed at the end of the day. Then you have the active transport, and this active transport, this is a diagram of active transport, but here you can literally see that the active transport pump. So we talked about the channel protein, we talked about the carrier protein, and this active transport pump resembles a carrier protein, right? But the difference with facilitated diffusion and active transport is that it requires the input of ATP. So, and the next thing is that in active transport, the molecules move from an area of low con. So, a low concentration to an air of high concentration, right? So this is the reverse, because in simple diffusion, facilitated diffusion, osmosis, they all moved from an area of high concentration to an area of low concentration. But in this situation, these molecules are moving from an area of low concentration to an area of high concentration, right? So it's basically almost like the opposite against a concentration gradient, right? It requires input of ATP. So now this is the definition. So active transport is movement of molecules or ions across a cell membrane from a region of low concentration to a region of high concentration against the concentration gradient, right? And it requires cellular energy in order to achieve this movement. That is why ATP is here. ATP meaning adenosine triphosphate, right? That's ATP. And an example of active transport would be our glucose, right? So earlier we said that glucose could go across the membrane via facilitated diffusion. Well, it can also go across the membrane via active transport, right? And active transport is also an endergonic reaction, which means that it uses energy and is also moving away from equilibrium, right? And it's going up against the concentration gradient, right? So that's basically the summary of simple diffusion, facilitated diffusion, osmosis, and then active transport. But the main thing is just to learn the phospholipid bilayer, because this is the bilayer. These phospholipids are what make up these are like the primary stuff, right? So lipids, proteins they play a huge role in our phospholipid bilayer, which I'm talking about the membrane. And in some within this diagram is a very simple diagram, but usually in between our phospholipids we usually have like cholesterol. On top we would have like our glycolipids or glycoproteins, and like a sugar chain, mean like carbohydrates. So yeah, but the main thing is just to know that this is hydrophilic head, hydrophobic tail, and because of the hydrophobic tail, some stuff cannot get across the membrane, and that's why they need methods like active transport, facilitated diffusion to get them across the membrane. Osmosis is also very much passive. It does not require ATP. The only one that uses ATP is active transport. The only one. Right, so anyway, so this is the end of the biology series. This is the end of diffusion, osmosis, facilitated diffusion, and also active transport.